Hi, my name is Dr. Adele Hosseini. I'm the Chief Scientific Officer at BOD Australia, and I'm going to talk about the key role of pharmaceutical standardized extract in medicinal cannabis. New medicines are being developed all the time, and historically, drugs have been originated from nature. Many drugs come from parts of the plants and microorganisms. One of the most common drugs available today is aspirin, that we are all very familiar with, and it comes from willow bark. The use of the salicylic tea to reduce fever goes back to around 400 BC, that people were using the willow bark extract as a medicine to reduce their inflammation, pain, and fever. But it wasn't until 18th centuries that pharmacists started to use it. And then in 19th centuries, it became a drug that is available as aspirin as we know it today. The other very well-known medicine that we are all very familiar with is the opioids, that it comes from opium poppy, and it's originated in 18th century. Then again, another one is that digoxin tablets, or digoxin in general, that it comes from foxglove plants, and it's from 1930s that and became available in 1930, and it again is originated from plants. So you can see that it's quite common for today's medicine that we see that they are coming from plants that now it's been extracted, developed, became pharmaceutical products, and people using it all around the world. Most recently, we have few chemotherapy agents that's also coming from plants origin. And the examples of them are vinblastine and paclitaxel, that they are all, both of them, it's been only the past 50 or 60 years that it's coming to the market. So medicinal cannabis is exactly the same. So medicinal cannabis needs to meet the same standards of quality, effectiveness, and safety expected of any other prescription medicine. What we see in normal medicine is the quality assurance of the medicine. In other words, each time that we use the medicine, we expect the same effect. In plant-based medicine, we need to have the reproducible chemical profile with minimum batch-to-batch variation. That's actually that's what we mean by extracts standardization. That will ensure the quality and safety, and ensures the consistency in efficacy, potency, and safety for the patients. So standardization of the extract is important for doctors for their confidence. Quite rightly, doctors want to know exactly what they give to their patients and what its effects are. And it takes a lot of time and effort for them to become used to prescribing cannabis. The reproducible chemical profile of a standardized medicinal cannabis will enable doctors to monitor the dose and effectiveness in the same way as other medicines that is used. Also, it applies to the pharmacists, who are the primary, and ultimately they are the one who are handing the products to the patients. So from patients' perspective, there is no need to say that the standardization, it helps with the efficacy and safety. And later on in the next few slides, I'll show you the impact that it's got specifically on the efficacy. For the researchers, it will make sure that the research that they are doing, it produces the robust data that can be used by industry in general, and it will lead to development of the products that is reliable. So in manufacturing also, the stable supply and reproducibility is really important factor. So why the standardization is so important in medicinal cannabis? The plant cultivars give rise to unique constituents and characteristics. For example, looking at this image, you can see a brassica plant. It's a same plant species that depends on the cultivar, it will produce different things and different characteristics. Kale, cabbage, broccoli, and cauliflowers, they are all coming from the same species, which is Brassica, but distinguishedly different. Another example that is quite familiar to all of us is the citrus, that you can see that even though the family or species of the plants is the same of citrus, but depends on the cultivar, we can see the difference between lemon, orange, lime, and grapefruit, for example. So it's the same thing that applies for the medicinal cannabis as well. Even though the plants is cannabis, but depends on what cultivar is used, it will produce different plants that it would have different constitutions and different characteristics. The most two common one is sativa and indica. That's pretty much used in industry. So cannabis doesn't have a fixed chemical or molecular compositions. A cannabis plant contains of hundreds of different substances, and each individual plant is different in terms of their composition. The THC and CBD are the most common one, but as we all know, there are hundreds of other chemicals or constituents is available that makes up of the plants and make the products available. Like we have about 70 of different cannabinoids that each of them, some of them is called as minor cannabinoids, and they all contribute to the effects and efficacy of the products that we are used as a medicine. In this few analytical or HPLC peaks, what we see is that 3 different cultivars which have unique cannabinoids profile, making each of them a unique products in their biological activities. So clearly, each of these peaks shows different cannabinoids, and what you can see is there is distinguished difference again between each of these cultivar, making these 3 plants that comes all from cannabis, but from 3 different cultivar, making it different medicine in a way. Cannabinoids often works well together and they produce a greater effect. That's also called as entourage effect. Entourage effect is when is the variety of inactive metabolites and closely related molecules markedly increase the activity of the primary endogenous cannabinoids. It's very well known that botanical drugs often are more efficacious than their isolated components. And we all know that the single molecules remain the dominant model in pharmaceutical development, but the concept of botanical synergy has been demonstrated that the pharmacological contribution of the minor cannabinoids and cannabis terpenoids to the plant's overall pharmacological effects. So with the new technologies and speed of science and research, standardization of the plant is now becoming a lot more achievable, and we can overcome some of the problems that we have with batch-to-batch variations. And therefore, the effect of the medicine is going to be a lot better and more reliable. A randomized controlled study in patients with pain also done and showed that entourage effect, that when they use the THC as an isolate in comparison to the whole plant extract that also contain THC, the results of the impact or the effects that the whole plant had on pain was significantly better than the isolate. The way that it works is the drug works is just they bind to the receptors in the body, the same way that the key will open a lock. So it's the same way as the drugs binds to the receptor in order to be effective. So when it comes to cannabinoids, the body does have cannabinoids receptors, and this endocannabinoid that is refers to endocannabinoid system, there will any products like a CBD or even the whole plants medicines or cannabis that we are using, it binds to different receptors, and all these minor cannabinoids binds to different receptors, and that's how it produces that entourage effects that we are talking about. There are a number of factors that determining the contents of cannabinoids. That includes the plant species and plant genetics that we talked in the previous slides. But controlling and monitoring the conditions under which the plants are grown, it's really extremely important. The harvesting also, it's got a significant impact on the quality of the products and also the contents of the cannabinoids. And storage of the final part of the extract is also very important. So it must be relatively small changes in cannabis composition as it has significant effect on the medical properties of the cannabis, as we discussed previously. So even the extraction method that is used, it has an impact on the effectiveness of the medicine or biological effect of the extract. The recent study that is just recently in 2021 was published by a group. They found that they actually done 4 different methods of extractions, and they evaluated the in vitro biological activity on human colon cancer and normal colon cells. Overall, this study indicates that variation in extraction method can lead to different compositions of the final extract composition and thus the different biological effect. The concerns that is around the quality of medicinal products is not just around the variabilities that is in cannabinoids contents, as we've been talking so far. The contamination with pesticides, with microbiological agents or mycotoxin, and contamination with the heavy metals and also the solvents. There are also some of those concerns that is available when we talk about the quality of the medicinal cannabis. To ensure the standardization of the cannabis, we need to make sure that the products are compliant with industrial standards for hygiene and safety, and most importantly, with the GMP or good manufacturing practice. Also, a good management system and good agricultural practice is very important, and we need to make sure that all the medicinal cannabis are compliant with that. And independent testings always has to be done to ensure that the products are free from contamination such as pesticides, heavy metal, and microbiological things as well. The products that currently available in the market around the world, some of them or most of them are not quite standardized. So lots of studies been done in different countries to just demonstrate that. And I'm going to show you a few of these studies that's been done in different countries. The first examples is the survey that's been done in Australia on I think 38 products that was available and was used by families with children with epilepsy. So what they've done is that these families been interviewed and the products that they were using were analyzed for the contents of the cannabinoids. The samples were rated by families as effective or ineffective. So the results of this survey showed that there was high variability in cannabinoids contents and profile of cannabis extracts rated as effective, with no clear difference between extracts perceived as effective or ineffective. Most samples contained low concentration of CBD, which was lower than what it was on the label, with THC amount present in them, whereas it shouldn't have been there. So these findings highlight the profound variation in the non-standardized cannabis extract being currently used in Australia. Similar results shown in the reports that's been done in the UK. That what they've done, they tested 30 products that is on the shelf available currently, like in 2019. Only 38% of the products were within 10% of the advertised CBD contents. 38% has had less than 50% of the advertised CBD contents, basically had like a half amount of the CBD that they said that they have. One product had 0 CBD in it, and almost half of the products had measurable amount of the THC or CBN in them, making it illegal products from the regulatory perspective. One sample that had 0 cannabis content, that was actually a premium products that were retailed for £90 in the UK. And one product, the amount of the ethanol was qualified as alcoholic beverage. In 7 products, heavy metals and solvents were also being seen. Exactly same type of the study is being done in the US as well in 2017, and the results is almost identical, that only 26% of the products had accurate concentration as it was labeled, 36% had less, and 22% contained more. And other cannabinoid concentration were really very low. So the solution is that the best choice is use of the pharma grade products to ensure the quality and reproducibility, and also the trust that the patient and a doctor could have in the medicine that they are using. Example of the partnership to focus on pharmaceutical standardized extract is the partnership between Linnea and Bud Australia. Linnea is the producer and manufacturer of a standardized cannabis extract based in Switzerland. And Bud Australia is an Australian company listed in an Australian stock exchange. And their mission is that to innovate and lead in R&D for delivery of the premium, proven and trusted products to patients and to doctors. The products that we have available currently in Australia is a pharmaceutically graded, GMP compliant medicabilis that it comes in 2 different forms. One is isolate and one is a full spectrum. And as I said, that this is pharmaceutically graded, partnership with Linnea, that they providing the API, and then we are producing the finished product and making it available to doctors. So currently this product's being prescribed by doctors in Australia through the TGA approval, and we had over 8,000 prescriptions, and the results, it's phenomenal with the satisfaction that we get from patients and the quality that from batch to batch that they don't see any differences, just because of we know that we are sure that this is GMP and pharmaceutical grade product. Okay, that's all from me. Thank you very much for your attention.